Hi, my name is Lindokuhle Magagula and welcome to my poster presentation titled Synthesis of Fluorescent Nitrogen-Doped Carbon Spheres from Corncob Residue for the Detection of Iron 3+ in Aqueous Solution. One of the major environmental issues is the contamination of water bodies by different industrial effluents. Among these industrial effluents are heavy metal ions such as iron 3+, which is one of the major contaminants of concern. Iron 3+ plays an essential role in living organisms. However, high concentrations of iron 3+ can have detrimental effects to both the environment and living organisms, which is why different methods have been developed for the detection of iron 3+, such as fluorescence quenching. In this study, fluorescence quenching will be utilized for the detection of iron 3+ in aqueous solution using nitrogen-doped carbon spheres that were synthesized from cellulose nanocrystals derived from corncob residue. These nitrogen-doped carbon spheres were synthesized using a one-step microwave synthesis at 180 degrees in 10 minutes. This was then purified and stored in the refrigerator for further use. From figure 3a, we can see that there's nitrogen that is present in our material. Therefore, it confirms that our carbon material has been successfully doped using nitrogen. As we can see from the UV vis, two peaks were obtained which were attributed to the pi to pi star transition and the N to pi star transition. For PL spectra, different excitation wavelengths were utilized to get the emission spectra, which is the one that is recorded. pH stability of the material was conducted. As we can see from this graph, the material tend to be relatively stable between the pH of 4 and the pH of 10. The pH value of 7 was utilized for further studies. The sensitivity of our nitrogen-doped carbon spheres towards the detection of iron 3+ was determined by preparing different concentrations of iron 3+ and incorporating them into the nitrogen-doped carbon sphere suspension. Now you can see that the intensity tends to decrease with the increasing concentrations from 0. This is because when the concentration is increasing, it tends to form a complex with the surface functional groups, therefore limiting the fluorescence emission of our carbon material. Results were then utilized to plot a straight line with the concentrations that are ranging from 0 to 500 micromolar. The limit of detection calculated using this line was obtained to be 70 nanomolar, which is comparable to previous studies. Selectivity studies were then conducted to determine if our material can detect iron 3+ in the presence of competing metal ions. As we can see, the red represents with iron 3+ and the other metal ions, and we can see that the intensity of our material with the iron 3+, the different metal ions with iron 3+, tends to decrease drastically. This is because our nitrogen-doped carbon spheres are highly selective to iron 3+. Therefore, we can conclude that highly luminescent nitrogen-doped carbon spheres with a high degree of functional group sensitivity and selectivity towards iron 3+ detection were successfully fabricated from corncob-derived cellulose. I would like to thank everyone.